Brought to you by Curious Marineland. So in this video, what I'm going to talk about is not everything here, but I'm going to link it into a second video. So I won't talk about hemopoiesis. I want to mainly focus on red blood cell production and how you produce hemoglobin and how you recycle it. And then later on we'll link that back into these two aspects of it. And we'll also get into some deficiencies. So we'll talk about the pathology after we talk about the normal physiology. So the pathophysiology. Again, brought to you by Curious Marineland. So let's first look at this. And this is going to be a picture you're going to see through a couple videos. So starting with this cell, this can eventually result in our big three cells that we talk about. The red blood cell, the platelet, and then all these that are called white blood cells. There's actually a couple of them. This actually has two types, T and B cell. So let's talk about the function of a red blood cell. The only one of these cells that doesn't have a nucleus. It's especially produced. Red blood cells only live to be 120 days, and they go through a lot of activity, and they're bouncing back and forth from going through the fluid with the plasma. That's the liquid. It's a formed element, and it's going to be banging into the walls of those capillaries. So some of them get damaged. So they only live to be about 120 days. So we've got to constantly replenish them. All right. And so we said it's anucleate, so it doesn't have a nucleus, but it's laced with this molecule called hemoglobin. So first, it's got two parts. You got the heme part and the globulin part. So this right here is the heme, and then see this whole thing here. That is the globular molecule. It's a polypeptide chain. Now with the heme group, embedded in it is iron. All right, if you're iron deficient, you're going to actually have some issues with the oxygenation of your blood because of what iron does. So let's look at the next slide. Now I have some stuff written here. Hemoglobin, what it does is this is what it picks up the oxygen. Now there's millions of hemoglobin molecules just in one set of. If you watch some blood, you're going to see a whole bunch of blood flowing through. You're watching millions of hemoglobin, and they're contained in the plasma membrane of the red blood cell, and it is laced with hemoglobin. It's so much laced with hemoglobin that it doesn't have a nucleus. It pops off the nucleus when it's being generated. That's for another topic. Okay, so let's advance to the next slide here. Move myself out of the way. So again, there's the heme part, which is these little circled parts, and then there's the protein part, which is, well, it's a protein. There's the globular part. Now what I want to point out about this right here is when it has oxygen, it doesn't have oxygen. All right, oxygen is not the thing that makes red blood cells red. Red blood cells are red with or without oxygen, except they're bright red or dull red. So all right, this is a bright red. Imagine this being oxygenated blood. See how now when it kind of dies down, you'll see it's with deoxygenated blood. So all that's imagine the oxygen being that's a red blood cell that has oxygen. Okay, this is a red blood cell without oxygen. So it's either less red or bright red. Now, if you remember your chemistry, remember oxidation and Leo-Gur. See, when oxygen attaches to the iron, it takes an electron from it. When you lose an electron, you increase your positive charge. The oxidation number increases. So this is with oxygen, and so without, and then imagine it being bright as it was before. It's not working now. All right. Now it is bright. So imagine this being oxygenated blood, and this is being transported throughout all the arteries to the capillaries where they're going to drop it off and pick up carbon dioxide. Okay, so a quick little thing about the lifespan. I mentioned that it's 120 days. Basically, about 1% of your red blood cells are replaced every day, and you have basically 3 million new red blood cells are circulating all the time. Okay, so let's talk about how do we actually recycle this hemoglobin. If it only lasts 120 days, unlike the federal government, the body is an amazing thing, and it can recycle and replenish and use things efficiently. Sorry about that jab, but they deserve it. Okay, so red blood cells, it mentions here, okay, so the kidney is going to be involved. The liver and the spleen are all involved, and the bone marrow. Bone marrow is where you're going to produce new red blood cells. So there are a whole bunch of enzymes. I'm not going to go into all of these, but let's look at this. The steps to recycling hemoglobin. So this is a cell that is at its end. It's going to die, and it doesn't just poof. We break it down all the parts, and we do something with it. So I'm going to come back to this picture, this slide here, but let's look at what's going on. There's a couple terms here. So yeah, hemolysis, that's the cell being broken up. So we're here. So hemopoiesis produces the red blood cells, and over the span of 120 days, about 90% of it works efficiently, and it's going to be retired. 10% gets damaged and retired. So you got a white blood cell called a macrophage. It's a phagocytic cell. It's going to come through and swallow and engage it. So here's the red blood cell. It gets broken into its amino acids and the heme part. The amino acids are the polypeptide chain making the globulin part of the hemoglobin. The heme part is right here. So what happens is you got a bunch of molecules that pick stuff up. So what I'm going to do is I'm going to come back to this, just pause the video for a second, and when you're looking at the picture, see if you can pick up these three molecules, because they're very important. So as this says, the globular proteins are broken into basic amino acids, and they'll be used wherever you need amino acids. Okay, the biliverdin is the next step. So when you take the iron off, it becomes biliverdin. Biliverdin is going to be converted into bilirubin. This is that orange yellowish color. Verdin is going to be greenish. If you ever have a bruise, it can be like greenish, and then when it starts turning orange and yellow, you're in the process of watching this conversion there. All right, now the liver is going to absorb the bilirubin, and it's going to become part of bile. Now some of that you get rid of. If you have excess of it, you can end up becoming yellowish and becomes jaundice. Now, what happens to the iron? See, the iron was stripped off, and it gets picked up by transferrin. So let's go back to this. If we're following the steps, if I were to number this, so this is kind of like the first step is split hemoglobin in half. Now look, the iron gets stripped off. The iron gets into the bloodstream by this molecule called transferrin. Where's it going to go? It's going to go back to where the new red blood cells are going to be produced. The biliverdin, some of it gets converted into another form called bilirubin, and it's going to go to the liver. Now, the excess, watch what happens to the excess. Some of it is going to, so here's the liver, it's going to go through the bile duct into the small intestines, and then go to the large intestines. Now, some of that is going to, now what I should show you, imagine some blood vessels that go right from the large intestines and the small intestines to the kidney. So some of this is excreted through feces, through waste, right here, and then some of it's going to be eliminated through urine. Now the rest of it, now it says hemoglobin is not phagocytized; they're broken down into polypeptide subunits. Basically, the stuff that's not, when it says broken down, the stuff that gets absorbed here, they're all going back to the red blood cells. All right, so that's hemoglobin being broken down. And this is the key thing. So my next video is going to say this stuff gets over here, and how do you get back to new red blood cells, and how do you get them into the blood circulation? So this hemolysis is red blood cells breaking in the blood vessels that gets picked up by macrophages and carried down to the liver. So, all right. So let me just check my time here. Okay, I think we can talk about what happens when you don't have enough hemoglobin, or you don't have enough red blood cells, and that is actually anemia. So let's talk about that. What exactly is anemia? It's a reduction in the oxygen carrying capacity. It can happen two ways. Now, low hematocrit, that's a way of measuring it. You can have low hemoglobin in the red blood cells, or you can take the red blood cells number and cause them to go down. So there's two ways of looking at it. So take a look at this. This is sickle cell anemia. So here's red normal, the normal physiology. This would be the pathophysiology. That's a term that me and my colleague have been using a lot, and I'll elaborate on some other videos with that. So this is clogging up. The red blood cells are not able to make it through, and it is a reduction of red blood cells, which is a reduction of its function, which is to transport oxygen. Anemia is less oxygen getting to the systemic system. Okay, so if I were to do it like this, anemia basically there's really two areas. Either something affects the iron, which affects the heme groups, so that the hemoglobin cannot bind up to oxygen. The other one is basically loss of numbers. This could be hemorrhaging. You lose blood, you lose RBCs. So these are kind of like the two methods. Now over here, this is the opposite. When you increase red blood cells, that's hemopoiesis is basically just the production of blood cells. That's another video. I'm going to skip talking about this. But so these are the two extremes: decreased red blood cells, decreased RBCs, and increased. So let's talk about this, and then we'll briefly mention that. So I'm not going to talk about every single one of these, but if you were to go through a number of these, each one of them would be either number 1 or number 2 of the category. So, all right, so like iron deficiency, folate, that affects the cell division. That's interesting, and that causes that cell division, so that affects the red blood cell production. Pernicious anemia is caused by a lack of nutrition, the B cell. That's one of the things, lacking of vitamin B12. Hemorrhage, that's this, you lose the number of our red blood cells. They go down. It's supposed to be arrow red blood cells going down. So let's see what else. Now, iron deficiency, basically you don't reabsorb that iron. If you go back up to here, so that's something that targets this. So these are all pathophysiologies that are all under the umbrella of what we call anemia. All right, and then this is hemolytic. That's what hemolytic. That's like a hemolytic disease. Like Ebola is a hemolytic anemia in all essence. Basically, the red blood cells are ruptured. They're destroyed. They're hemolysis. Lytic means lysis or lytic. So here's the other side of this. So we go back up to here, just to expand on what is this polycythemia, right? So that's increasing red blood cells. Now, if you increase too much, that could be uncontrolled mitosis, uncontrolled number. That could be a type of cancer. But a lot of athletes will go train. The Olympics, they train in the Colorado, so high altitudes causes an increase in hemopoiesis, and that will mean you have more red blood cells, you have more oxygenation to all your muscles, and you can perform better. All right, we'll talk about erythropoiesis later. Okay. Now, I mentioned the hemolytic one just as just a quick little side note. I think I'm going to run out of time, so I should talk about it later, but this is something that's related to the blood type of the mother. So it's called erythroblastosis fetalis, and probably I can let my PowerPoint do the talking. I got a shorter version I use for baby bio. So in all essence, what happens is if the mother is negative, blood types ABO or the plus minus. This is talking about the plus minus. If the mother is positive, if the mother's negative and her first child is positive, nothing's going to happen to that child. But her second child, she's going to make antibodies against it. And then that baby basically is going to be targeted. It's going to be spontaneously aborted or miscarried. And there's treatments for it. Basically, Rogam is one we use, which it's an immunosuppressant. It prevents the mother from targeting her own immune cells, killing the child. So this is a picture my students got in class, so it's a little more detailed, and I expect them to read through that and do a diagram about it. It's basically your anemia by result of ruptured cells. So most people don't consider it anemia. They consider it another category. All right, so that's pretty much it. This is my video on hemoglobin and anemia. Now, I want to mention these two and close out with so this is red blood cell. Anemia is red blood cell. Polycythemia. So this is too little, this is too much. These two are all dealing with white blood cells. So this is just pause the video if you need to read this. So this is like the white blood cell version of anemia. Okay, this is like if you've ever been to the doctor and they say, all right, well, you got elevated elevated white blood count. That's what this is, the leukocytosis. Unfortunately, if it's a continuation and continues to happen, that actually can lead to a cancer that we call leukemia, which is uncontrolled growth, uncontrolled mitosis. That's what cancer is, uncontrolled mitosis of the white blood cells. One last one is we left off platelets. Platelets, low blood count for them is actually called thrombocytopenia. Remember, a red blood platelet is called a thrombocyte, so if you thrombocytopenia, which means they're going to have issues with clotting their blood. Not full-on hemophilia, which is another pathophysiology that we'll look at. All right, brought to you by Curious Maryland. And so I'm going to go to my next video, and it's going to expand on the production of all the blood cells. All right, thanks for watching.